Imagine you're born with a book inside you. Not a storybook, not a diary, a manual. Every page of this book decides something. How tall you'll be, what your eyes will look like, how strong your heart is, even what diseases you might get. For most of human history, we knew this book existed, but we couldn't read it. Doctors could see the effects of disease, but not the instructions behind them. Scientists knew DNA carried information, but they didn't know what that information actually said. So in 1990, something bold happened. A group of countries came together and said, let's stop guessing. Let's read the whole book. That mission became the Human Genome Project. The goal was simple in words, but huge in reality. To read every single letter written in human DNA, all 3 billion of them. Think about that. 3 billion letters A, T, G, and C spread across 23 pairs of chromosomes inside every cell of your body. Those letters are not random. They are instructions. Instructions for building muscles, for growing nerves, for repairing skin, for fighting infection. Scientists had to cut DNA into millions of small pieces, read them using machines, and then use powerful computers to put the whole thing back together, like rebuilding a shredded book page by page. It took years. It took thousands of scientists. It took countries working together. But they weren't doing it out of curiosity alone. They did it because behind every genetic disease, every inherited cancer, every unexplained disorder, there was a broken sentence somewhere in that book. And if you can find the broken sentence, you can start fixing the problem. That's what the Human Genome Project really was. Not just a science experiment, but humanity finally learning how to read itself. After deciding to read this book of life, the next question was obvious. And why would anyone even try? This wasn't a small lab project. Reading 3 billion DNA letters was going to take machines, money, and years of work. Many people at the time said it was impossible. Two main groups stepped forward. One was the U.S. National Institutes of Health, the part of the government that deals with human health. The other was the U.S. Department of Energy, which sounds strange until you know the reason. After World War II and nuclear testing, scientists were worried that radiation might damage human DNA. So, the Department of Energy wanted to understand what normal human DNA looks like so they could detect what radiation changes. So, these two groups joined forces. Soon other countries came in. Britain, Japan, Germany, France, China. It became a global mission. But there was a twist. At the same time, a private company said, we can read the human genome faster and we will own it. That meant human genes could become private property, something to be bought and sold. The public Human Genome Project made a firm decision. The human genome belongs to everyone. So, this was no longer just a science project. It became a race not just to finish first, but to decide whether the basic code of human life would be free or owned. And that's what pushed the project forward with full force. Now the mission was set. The book existed. The team was ready. So the next question was, what exactly were they trying to read? Inside every one of your cells are 23 pairs of chromosomes. These chromosomes are made of long strands of DNA. And DNA is written in just 4 letters, A, T, G, and C. Those 4 letters form words called genes. And those genes tell your body how to make proteins, the things that build and run everything in you. The Human Genome Project had 3 big tasks. First, find every gene in the human body. Second, read the exact order of those DNA letters. Third, map where each gene sits on the chromosomes. But this was not like reading a normal book. DNA had to be cut into millions of tiny pieces, copied again and again, read by machines, then reassembled by computers. Imagine tearing a giant book into shreds, scanning every piece, and then asking a computer to put it back in the right order. That's what scientists were doing on a scale never tried before. And this is why the project had clear goals. Not just to read DNA, but to store it, analyze it, protect it, and make sure the knowledge could be used by doctors and researchers everywhere. This was the moment biology stopped being small scale and became big science. Now, something unexpected started to happen. As the pages of the DNA book were being put together, scientists began to notice patterns, and some of them were shocking. For a long time, people believed that humans must have an enormous number of genes. After all, we are complex. We think, feel, create, and suffer from thousands of diseases. But when the Human Genome Project counted them, the number came out to only about 20,000. That's not much more than a mouse. It meant something important. Being human is not about having more genes. It's about how those genes work together. They also discovered that all humans are almost identical at the genetic level. More than 99.9% of your DNA is the same as every other human on Earth. So skin color, height, eye shape, even disease risk, all of that comes from a tiny fraction of our DNA. And then came the medical breakthrough. Scientists could now look at the genome and say, this gene is linked to breast cancer. This one affects heart disease. This mutation causes a child to be born with a genetic disorder. For the first time, disease had a map. Doctors were no longer just treating symptoms. They could see where the problem began, inside the code itself. That is when medicine quietly entered a new era. Once the human genome was read, medicine was never the same again. Before this project, doctors treated people based on averages. If a drug worked for most people, it was given to everyone. If it failed, no one knew why. But after the Human Genome Project, doctors could look at a person's DNA and see differences that mattered. They could predict who is more likely to get cancer, who might develop diabetes, which medicine will work, which medicine might cause harm. This is called personalized medicine, treatment based on your own genetic code, not guesswork. The project also made genetic testing possible. A simple blood sample could now tell parents if their child carried a genetic disorder. Cancer could be detected early, even before symptoms appeared. And then came something even bigger. If you can read DNA, you can also edit it. That idea led to tools like CRISPR, where scientists can fix broken genes, like correcting a spelling mistake in a sentence. The Human Genome Project didn't just explain disease. It gave humanity the tools to fight it at the root. Medicine stopped reacting and started predicting. The Human Genome Project didn't just read our DNA. It changed how we understand life itself. For the first time, humanity looked into its own instruction book and learned how to fix its mistakes. And this was only the beginning. The book is open now, and the future of medicine is being written from its pages. If you found this video helpful, don't forget to like it, share it with your friends, and subscribe to the channel for more simple and clear explanations of biology. Keep learning, stay curious, and I'll see you in the next video.